When we talk about genes, we often think of the set code in our DNA that determines our eye color, hair color, and many other characteristics. However, the world of genetics is much more complex, and this is where epigenetics comes into play. Epigenetics deals with changes in gene expression that are not due to changes in the DNA sequence. In other words, while the DNA sequence forms our genetic basis, epigenetics determines how this genetic information is used. A better example, cells in the same genome can take on completely different identities. For example, a liver cell performs completely different tasks than a muscle cell, even though their cells occupy the identical DNA matrix. Therefore, genes can be selectively switched on and off and have various effects on cells. The following modification methods play a role. DNA methylation. The attachment of methyl groups to DNA can cause genes to be switched off. This methylation changes the structure of the DNA and prevents transcription factors and other proteins from binding to the DNA to initiate gene expression to produce proteins. Histone modification. Histones are proteins around which the DNA is wrapped in the cell nucleus. Chemical modifications of the histones, such as the attachment of methyl or acetyl groups, can change the structure of the chromatin and regulate gene activity. RNA interference. In RNA interference, small RNA molecules, such as microRNAs, bind to the messenger RNA and prevent it from being translated into proteins. In this way, genes can be selectively switched off. CRISPR-Cas9. The CRISPR-Cas9 system is a method of genome editing that can be used to selectively make changes to the DNA to switch genes on or off. The DNA is cut at a specific site and replaced with a new DNA fragment containing the desired changes. Through these modifications, activities of genes can be increased, reduced, or completely switched off, which is quite interesting. Now the question arises, what reinforces something like this? Research has shown that environmental toxins and pollutants in the air, water, or our food can lead to epigenetic changes. Certain nutrients, such as folic acid and vitamin B12, play an important role in DNA methylation. An unbalanced diet that leads to a deficiency in these nutrients can thus affect gene expression and thereby our health. Also, foods containing bioactive substances, such as broccoli or green tea, can induce epigenetic changes and influence the activity of genes involved in the development of cancer. Our life experiences and psychological state can also influence epigenetics. Stress, trauma, or positive experiences such as love and security can affect the expression of genes involved in the regulation of stress hormones and the function of the immune system. Thus, a traumatic experience can lead to long-term changes in gene expression that affect our stress response and our mental health and have biological effects on our body. In summary, epigenetics shows us that our genes are not just a set piece of information, but rather a dynamic structure that can be influenced by a variety of factors. The insights from epigenetics open up new perspectives and possibilities for preventing diseases and actively promoting our health. They also show us that we can have a significant influence on our genes, and thus our well-being, through our lifestyle and psychological attitude.
If you don't want to miss further videos about interesting topics like this one, subscribe to our channel and activate the bell to be notified when we publish new short videos. Thank you for watching.